Okay, so this is lecture 16, and we had been looking at the organelles in the last class. And again, if we recall the origin of the organelles, the nucleus was thought to come about from an invagination of the membrane, and as a result, it has a double membrane around it. The mitochondria shown in this example, but chloroplasts, the same thing would have happened. They were thought to be a relationship between a bacterium and an ancient eukaryote. And it may, in fact, have been a parasitic relationship because the mitochondria is thought to be related to a group of what we call intracellular pathogens, which are bacteria which grow inside cells, called rickettsia. And here we can see the mitochondria is taken up forming this endosymbiotic relationship, which allows this new cell to produce a whole lot of ATP. And we didn't show the example of it, but chloroplasts likewise were thought to have been taken up by an ancient eukaryote. And chloroplasts have the ability to photosynthesize, so to take light and to produce ATP from that. So this is something we'll come back to a few times when we're looking at endomembrane system. And this is this idea of the topological relationships between compartments. So what we've done here is this little square is just representing the outside world. So the extracellular space. And so that's shown in this pinkish color. And what we can see is the kind of chemical equivalents include that space surrounding the nucleus, which becomes the endoplasmic reticulum, which is an organelle we'll be looking at today or Tuesday. The Golgi, lysosomes, endosomes, these all have a similar, what we call a topology or chemical composition to the extracellular environment. And we'll see why that's important in the coming weeks. In contrast, the gray represents the topology of the cytosol, and that's the same as what we see inside the nucleus because it has that weird gated transport. So inside the nucleus, we have the compacted DNA, which we call chromatin. That's the DNA associated with a large number of proteins. And we also learned that how that DNA is packaged is actually important for it and can affect its expression. And that was the field we called epigenetics. And so I think where we had left off was talking about the nuclear pore complex. Super cool transporter, and it's unique because it allows for facilitated diffusion of small molecules, meaning they'll just flow down a concentration gradient. In contrast, larger molecules are selected and moved via active transport, meaning that there's an input of energy for the movement of those molecules. So again, if we go back to this idea of our GTPases, so we had kind of finished with this in the last class, and I'll draw it up quickly again here. So the GTPases exist in two conformations, so two different structures for the same protein. And in one form, in the on form, they're bound to GTP, so that's a G with 3 phosphoryl groups. And in the off state, they're bound to GDP, which is just G with 2 groups. And so where we got to was we talked about how we go from on to off, and we had discussed how we go from off to on. And so to go from on to off, I'm just going to draw these arrows in now. Okay, so to go from on to off, we have water comes in and orthophosphate, which is inorganic phosphate, comes off. And so that is this phosphoryl group. So water comes in and breaks that bond. All right? Hydrolysis. So now the GTPase is in the off form. To turn on, what it actually has to do is it actually has to kick, and I'll do it in a, So it actually has to kick that GDP out. And what has to come in instead, I'll do this in a different color, is a new GTP has to come in. So the GDP is not phosphorylated to GTP in place. So I always ask this on an exam. I'll ask it on your exam at some point this year. So the GDP gets kicked out. And what's important here is we have two states, so this can serve as a switching mechanism. Okay? So it can be in an on or off state, and that turns out to be really important for signaling pathways. And in the example we're going to see today, it's really important for helping the cell determine if the space it's in is the cytosol or the nucleus. The processes can be catalyzed, and the enzyme that catalyzes the hydrolysis is called the GAP for GTPase activating protein. And the enzyme, and so that's a GAP, and the protein that helps to turn it on is called a GEF for a guanine nucleotide. Exchange factor. Remember, GTP is a guanine nucleotide. And so those are enzymes that can catalyze that, and we can regulate the activities of those enzymes to determine how active they are. So the GAPs and GEFs are going to be important in our nuclear localizations and nuclear import and export, because the location of GAPs and GEFs are going to determine the state of our GTPase. Questions about that? So you're going to see this slide at least one, probably a couple other times, because the GTPases are really important. So we're going to look at nuclear import and export today, and then we're going to look at 3 organelles that are working in a related pathway called the endoplasmic reticulum, the Golgi, and lysosomes, and we may get as far as peroxisomes today as well. So on the left-hand side, we have import into the nucleus, and on the right-hand side, we have export from the nucleus. And we know certain things are specifically brought in and out of the nucleus. This is an example of active transport. These are bringing large molecules specifically across that double membrane. So the GTPase is a protein called RAN. Right? So we can see RAN here. And inside the nucleus, we have the GEF. So what that means is inside the nucleus, if there is RAN bound to GDP, that GEF is going to kick the GDP out and allow GTP to bind. Okay? So inside the nucleus, RAN is bound to GTP. In the cytosol, we have the GAP. So what that means is GTP will be hydrolyzed to GDP. Now we can see the importance of our little monomeric GTPase. This RAN will be bound to GDP in the cytosol, and that informs the system that RAN is in the cytosol. In contrast, when RAN is bound to GTP, it informs the system that it's inside the nucleus. All right? So now we have a way of determining which compartment we're in. And now we just have to kind of work through the nuts and bolts of the actual system. So on the left-hand side, we're looking at import, so bringing the specific targets into the nucleus. And this is mediated by what's called a nuclear localization sequence, or NLS. Nuclear localization sequence is found on proteins that will be imported into the nucleus. And then the nuclear localization sequence will bind to one of a family of proteins called importans. Okay, so the importans bind to the nuclear localization sequence, and that whole complex is imported into the nucleus. So what's the importance of RAN here? So RAN, in its GTP-bound form, will bind to the important. We can see this here. So the GTP-bound form will bind to the important, and that prevents the important from binding to its cargo. So when it's bound to GTP, the RAN will bind the important and prevent the important from binding cargo. Of course, we're in the cytosol, so the RAN will be in the GDP-bound form, and this can't bind the important. So in the cytosol, the important is free to bind to cargo, and it's brought into the nucleus. What happens inside the nucleus, of course, is that RAN is in the GTP-bound form. That's how our switch worked. So in the nucleus, RAN will bind to our important, we see this here, and that means the cargo is released. If we look at the reverse process, we have a nuclear export signal, NES, on the protein. And the nuclear export signal is telling the system to pull that protein out of the nucleus. This is also mediated by RAN, but in the opposite way. So in this case, the exportin can only bind to its cargo when it's also bound to RAN GTP. So exportin will bind to RAN GTP. That allows it then to bind the cargo, and the whole thing is exported out of the nucleus. What happens to that RAN GTP in the cytosol? It's exposed to RAN gap, which hydrolyzes the GTP to GDP, and now this can no longer hold on to the exportin. The exportin is released. The released exportin can't bind to the cargo, so the cargo is also released. Is there any questions about that? Yes?
What's the cutoff for the upcoming exam? Is the upcoming exam just going to the end of the nucleus, or?
So the next exam goes to the end of the nucleus. Yeah. Questions? Okay, so there's kind of an issue here. So I'm going to give you a few, maybe 20 seconds to see if someone can figure out what the problem with this whole system is. So if you look carefully at this diagram, there's a problem. It's a bit of a tricky question. Okay, does anyone see what the problem is? Yes?
Just yes, but is it because like in the left side of the slide where the GTP is bound to RAN, isn't that like not?
Oh, okay, so here where they're showing it bound to RAN, So this part they're just showing immediately after it's been exported. It'll still have the GTP, and then that'll quickly
They'll
be.
just immediately.
Yeah, pretty much immediately. But it is something to do with that sort of step. Yeah.
Is it that we run out of RAN inside the nucleus?
Yeah, look, the RAN's never going back in. We can see both the important and the exportant are cycling, but the RAN isn't moving back in. Good observation. And actually, this is mediated by another enzyme or another protein carrier. You don't need to know the name of it, but it's called the NFT2, and that mediates the import of RAN back into the nucleus. Because in this, both sides, we just have the RAN going out. So this is an example of a nuclear localization signal. Okay. And how do you determine that this is a nuclear localization signal? All right, so you've looked at 20 proteins that get imported into the nucleus, and you notice that all 20 of them have this same sequence at their N-terminus. Okay, so this is an N-terminal sequence. So how do we confirm that that's a nuclear localization sequence? Well, what we can do, and I'm going to dazzle you with some gene constructs again because we love that so much. So what we can do here is we have a gene of interest. And I'm going to call it NLP for NNP for non-nucleus protein. This is a protein that doesn't go into the nucleus. And we remember how this works, of course. We get translation starting at this +1 site, and we're going to make an mRNA that covers that, and that's going to get translated into a protein, okay? So what we do is we make a fancy new gene where before our NNP, we add in an extra little sequence that's our nuclear localization sequence. So now our mRNA excuse me. And so this is of course, it's a single gene. So you've made a single gene, but you've modified it so now your mRNA is a little bit longer and has that nuclear localization sequence at the start, which will be translated onto the protein. And what do we expect now to happen with our non-nucleus protein? If it's a nuclear localization sequence, what's going to happen to that protein? It's going to move into the nucleus. So we've modified the gene, we've made it a little bit longer, and we've added this sequence of the nuclear localization sequence to the N-terminus. We make another one, which is what we call a control, where what we do this time is we have our nuclear localization sequence. So we're making this slightly longer protein, but this time we introduce the wrong amino acid at one position. So now what we're making is an mRNA for a protein that has a disrupted nuclear localization sequence. And so if we look at our notes, which you have in front of you, we can see a great example of a non-nucleus protein. And what is that? Yeah, what's the protein?
Oh, I was asking a question.
Oh, sure.
So is the NLS, the bond on the site forwardly important?
Well, that's what you're testing.
Okay.
So the question was, is the NLS what the important binds to? And this is how you would test it. And this is how we can identify these. So we think it is. We want to know. So the first thing we need to do is see if this puts something into the nucleus. So we make these two little tags. What's a non-nuclear protein we can add this to that we can see? We can add this to GFP. So on the left-hand side of this, the sequence shown at the bottom is the putative, meaning the thing we are guessing is a nuclear localization sequence. Putative means you predict something is going to happen, but you haven't proven it. So this is, in this case, the putative nuclear localization sequence, proline, lysine, 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 arginine, lysine, valine. And we add it to our gene, so we're still making one gene, one mRNA, one protein, but this protein now is a little bit longer and has this sequence at the N-terminus. And if we look at where the GFP goes, it all goes, in this case, into the nucleus. We don't see any in the cytosol. We do the same experiment. We modify the gene. It's still a little bit longer, but we've disrupted it by swapping out one of those lysines for a threonine. Okay? And what we see now is that the GFP localizes to the cytosol, and the nuclei are actually blank, dark holes, because the GFP doesn't go in there. So now we've moved from calling these a putative nuclear localization sequence to calling this a nuclear localization sequence. And so again, we do this little bit of genetic engineering, in this case to test our hypothesis that this is a nuclear localization sequence. Pretty cool, eh? Any questions? So that will be the cutoff for Friday's exam. I will post some extra office hours for next Thursday. I just have to sort of work out my schedule. We have office hours on Monday from 1 till 3, if you have any questions. Don't forget to read the study tips that I posted. Again, there's a bunch of practice problems and things posted as well. Any questions about the nucleus? All right. So let's move on to the endoplasmic reticulum, which we know is basically sort of an extension of the nucleus. So it's continuous with the nucleus and was likely formed, again, through that invagination of the membrane. So it's important, it has a number of important functions. Yes?
Sorry, could you just spell out invagination?
Oh yeah, sure. Just like that. It's just like imagination, but going in. So if we're looking at the endoplasmic reticulum, as with all organelles, it has specific roles. One of them is that it serves as a storage space for calcium, Ca2+. And remember how we talked about how the cell has a very specific concentration of sodium and potassium inside it. So it keeps sodium low inside the cytosol and potassium high. And that turns out to be really important because the concentration of potassium outside is low and the concentration of sodium is high. Any of that ringing bells, hopefully? There's another trick that it does in that it makes an internal concentration gradient where the cytosol has very low concentration of calcium, But inside the endoplasmic reticulum, so an organelle within the cytosol, or in the cytoplasm, I guess, it's very high. So it can control, in this case, the concentration in both the cytosol as well as the organelle. And we're going to see that's really important for signaling later in the course. And there's one type of signaling where you may have heard about this before where it's really important. And does anyone remember what that is? It leads to a special kind of physical reaction. Anyone know? So this is how muscle contraction, I can see people there flexing their arms. So yeah, this is how muscle contraction works. We have a specialized type of endoplasmic reticulum called sarcoplasmic reticulum in muscle cells that stores high concentrations of calcium, and this allows for muscle contraction. It's where we produce transmembrane proteins. So those are proteins, remember, that span the membrane. And lipids are constructed here. So not all lipids, but most lipids. It's also where proteins are manufactured and modified, not only for transmembrane proteins, but also secreted proteins, things that are actually going to physically exit the cell. And so we'll talk about the endomembrane system a little bit later in the course, and that is the process of secreting proteins. There are two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth. They're all found within the same organelle, but they're on unique parts or spaces of the compartment, so you can physically separate the smooth and rough endoplasmic reticulum. The rough endoplasmic reticulum is named because it physically has a roughened surface. This is looking at a transmission electron micrograph. So of course we can see membranes and we can see ribosomes. And if we look at these, this is a very thin section through it. We have all of these sort of, that's not a good color to use. We have all of these kind of compartments. And that's showing the membrane of the ER. And then the whitish part is the space inside, which is called the lumen. That's the inside of the ER. And peppered along the cytosolic face of the ER, we see all these dots. Okay? See them all over. Those dots are actually ribosomes that are associated with that membrane. And the ribosomes, of course, are the little factories making protein. And so the proteins are made, not all proteins, but proteins that are going to enter into the ER are made right at the surface of the ER. The smooth ER, in contrast, this is important for our lipid metabolism, our storage of calcium. It's also an important step in the detoxification of many drugs. So drugs are generally designed to be hydrophobic. This allows them to move into cells. And one of the first steps when you're trying to get rid of a drug, one of the first things we do is we increase its polarity. And so we do this through a process called oxidation. And that's carried out by a group of enzymes called the P450 enzymes. Or that's one of the ways it can be done. And that's associated with our smooth endoplasmic reticulum. How do we separate them? This is kind of fun. First of all, we just separate the ER out. We can, yes?
Is there a higher concentration of smooth endoplasmic reticulum in like the liver or?
Yeah, so the question is there differences between the smooth and rough ER between different types of cells? Yeah, and if you go back to, we compared the pancreatic and the liver cells in the last lecture, if you actually look, you can see there's a big difference in the content of smooth versus rough ER for them. So that will be tailored to the functions of the cell. Exactly. So we isolate our ER, which is both types, and then what you do is you make these microzones. And basically what you do is homogenize, or just kind of shake or stir or blend these up. And when you blend them up, you're breaking the membrane, right? But the membrane reforms to make these little microsomes. And the microsomes are what the microsomes in fact are. If we were to take one and blow it up, we would see it's our lipid bilayer. Oops, that's not what a lipid bilayer looks like. So it's our lipid bilayer, okay? Inside will be our little lumen. And outside, if it's rough ER, outside it'll have the ribosomes stuck to it. And if it's smooth ER, it won't. Why, when you rip the membrane like this, does it reform little sort of vesicles or microsomes? Why doesn't the membrane just stay kind of ripped apart? Yeah.
Because it's favorable to form the layer.
Yes, it's favorable to form that layer because if you rip the membrane, you've exposed those hydrophobic regions. So they actually just seal up on their own when you tear it, which is super cool. So we can take this and we can do our equilibrium centrifugation or another type of density gradient centrifugation. And we can physically separate the microsomes from the rough ER because they're more dense. They have all those ribosomes stuck to them than the smooth ER. And so this can be used for studying purposes. If you're doing research on, say, just the rough ER, you're looking at the proteiny parts of it, you can do this and isolate microsomes. We'll go through this in more detail later in the course a little bit, but here's how the process works. And again, we're just going to give a pretty rough introduction here, and we'll show it in a little more detail later. Again, all of this will be on the second or the final exam, I guess. Wow, this year's really going fast. You'll be able to, we'll have covered it by the time we get to that. So what this is looking at is the blue line shown 5' to 3'. What do you think that might be? So that's our mRNA, right? The messenger RNA, which is going to be translated. The green thing is our ribosome, the big green blob. And then this here in green and red is this is the actual protein. And at the N-terminus of the protein, in red, we have this, again, a localization sequence, which is enriched in hydrophobic residues. So it's part of the protein, this red. It's translated. They're just indicating it in a different color because it's the signal sequence. So the N-terminus is the first thing made by the ribosome. So it's the first thing that emerges from the ribosome, right? So of that whole protein, the first thing that's going to pop out into the cytosol is that little red bit. And when that comes out into the cytosol, it's bound by a special protein. We don't have it on here. It's bound by something called the signal recognition particle. We'll discuss this, as I said, in detail later. And translation stops. It docks onto the surface of the endoplasmic reticulum. So this is the ER membrane. Oops. ER membrane. Here's the lumen. And of course, we know translation always occurs in the cytosol. So what happens is translation is stopped until the ribosome binds to this special thing called the translocon. Translocon. And then once it's bound to the translocon, translation resumes. And what this means is that as translation is occurring, the protein is moving through the translocon into the lumen. So we call this co-translational translocation. So because as translation is occurring, the protein is directly moving into the lumen of the ER. Okay. It's important to note translation, the whole process of translation is actually still in the cytosol, but the protein itself, and this is a key component here, the protein itself doesn't interact with the cytosol. It moves into the lumen. And in fact, that signal particle, that little red part, is cut off. So the green part, which is the mature protein, has never touched the cytosol. That's important for later. The other way that we can do this is post-translationally, so we can build the protein, we can unfold it, and we can pull it into the ER, and we can refold it. This is it in a little bit more detail, but not a lot more detail. So again, the signal is these nonpolar amino acids. Okay. The signal sequence was discovered using in vitro translation studies, meaning what they actually utilized was those isolated microsomes from the rough endoplasmic reticulum. And then, similar to what we did with the nucleus, we can add or modify the signal and see if those proteins end up in the endoplasmic reticulum. It's mediated by a special molecule called a signal recognition particle. And again, this is something we'll give more details from soon. This is a molecule made up of RNA and protein together. When the protein is translocated, it can be glycosylated, meaning we can add sugar to it. And those sugars will be covalently attached via an anomeric carbon. In this case, they get added to an NH2 group on asparagine. Remember asparagine at the end has this. So our linkage is through this to the anomeric carbon of our sugar. And so we call this an N-linked glycosylation, because what it'll end up looking like is this, and then it'll be attached to our sugar. Okay? So we call this N-linked glycosylation. It's still through the anomeric carbon. It's just we're not bridging through an oxygen, but rather through a nitrogen. The N-linked glycosylation only occurs in the endoplasmic reticulum. There is also an O-linked glycosylation that occurs in the Golgi, another organelle, the one we'll be looking at next. The way this works is super cool. This is showing how the sugar is manufactured. On your right-hand side, you have the cytosol. On the left-hand side is the lumen of the endoplasmic reticulum. And I'm not going to ask you to memorize how it's constructed. But what's super cool about this is that our sugar that's added, okay, so our sugar that's added is this. So this is made partially in the cytosol, partially in the ER lumen, and it's made up of 14 subunits, the same subunits. And it's made up of N-acetylglucosamine. Again, you don't need to know the composition at this point, which is in the blue, mannose, another carbohydrate, and glucose in gray. And this is the cool part. As that protein moves in to the ER, the transferase, the thing that's going to move this giant block of sugars onto the protein, is actually associated with the translocon. It's like being at a wild rock concert here. So the protein's coming in, a target asparagine is seen, and that whole block of sugars, all 14 of them, are attached to that asparagine residue. And then, if we look at the bottom, we can see we start to modify that sugar. So the initial block that's added, for all proteins that are end-linked, it's always the same. But if you were to look at the final secreted mature protein, you would see they're different because they undergo modification both in the endoplasmic reticulum and in the next organelle we see, which is called the Golgi. Questions? So the Golgi was first recognized, okay, I don't have the, what is it, 1898. And this is showing a picture of Golgi. He won the Nobel Prize, but not for the discovery of the organelle called the Golgi. He won it for something else. He first made these observations, and there's a beautiful drawing of his in the top left. He took his samples and he did this special type of stain which he called the black reaction, which sounds very wizardy and Halloween-y, so it's totally appropriate for this month. And so we used this metal impregnation, and these showed up. And like most people just didn't believe them. Okay? So it was thought to be an artifact of the staining. And remember, what's an artifact? It's something that you think is real, but it's not. It's just you're being misled by something. It's just to do with how the reaction or how the staining was occurring. It just happens to form these clusters that look like an organelle. In the 1950s, however, the presence of Golgi bodies, sometimes you hear it called Golgi apparatus as well, which I think is cooler, so I like to call it the Golgi apparatus. These were first recognized in the 1950s as, this is real, so 50 years later. And what happened in the 50s? Why could we suddenly confirm this? What do you think happened? Yes, electron microscopy made it possible to actually observe these Golgi bodies. The Golgi is a bunch of smushed up discs, kind of flattened discs that have a space inside them. And here is a really nice transmission electron micrograph showing them. And on the right, we have a depiction of them. Okay? And we can see then that they kind of in some ways look similar-ish to the ER, but instead of being one kind of continuous moving around, highly lobed organelle, these stacks are actually distinct. So you can think of this thing like a pile of pancakes. This is a major site of carbohydrate synthesis, and by that we mean more polysaccharide. So not the actual construction of the subunits, but putting those subunits together into oligosaccharides and polysaccharides. Proteins are modified in the ER, we saw that. They then move to the Golgi and they get further modified. And one of the things that happens is that we can get linking to the hydroxyl group, the OH, on a serine or a threonine, and that forms an O-linked glycosyl group. And so O-linked sugars are only added in the Golgi, N-linked only in the ER. And this is a smaller proportion. It's something like 10% of the overall glycosylation is O-linked and 90% is N-linked. This is an organelle that undergoes constant change in its shape and size. And so because of that, all of the resident proteins, those are proteins that are always associated with the Golgi. They're membrane bound, so they're not lost. And they're all single pass transmembrane proteins. And I think we'll end it there and we'll start it with the Golgi. Everybody have a great weekend. We'll see you on Tuesday.